All right, so it looks like everything is up and running and ready to go, so I will get started. Hi, my name is Sydney Preston, and I am a biology and public health major, and I am here to talk to you about my summer research that I did this most recent summer, which was mutations along the COVID-19 genome, and I looked specifically at Minnesota cases. So, as just a little recap of where the US stands as of the 23rd is when I grabbed this data. We are at a total of 8.5 million cases of COVID in just the US, which is a pretty large number. And in Minnesota alone, we are sitting at 129,916. And for deaths, we are at 2,304. And sadly, this is only on the rise. And again, this was taken on the 23rd, and today is the 28th. So the cases have definitely gone up, and deaths as well, which is a shame. So, as a little bit of a background on SARS-CoV-2, which needs no introduction, we're all fairly familiar with what it is and what it's doing. It originated in the Wuhan seafood wholesale market in Wuhan, China, in the Hubei province. The origins of the virus are still unknown. Scientists are guessing that it's a bat reservoir. Again, scientists are still unsure, but the viral genome itself shares 96% similarity between bats and humans, hence why they're thinking the reservoir is a bat, specifically horseshoe bat. Many hypothesize that the Chinese horseshoe bat, again, is the origin of the SARS-CoV-2 virus, as it was the origin for the earlier SARS-CoV outbreak in 2002, which I found in the Lau et al. 2005 article. Coronaviruses in general are very, very recombinant, which makes it difficult to ascertain the origin. And the type of virus that SARS-CoV-2 is beta coronavirus. There are 4 genera of coronaviruses, which overall are alpha, beta, delta, and gamma. I won't get too deep into that, but what makes them into the different genera is like their structure, the viral genome itself, and things that make the virus unique. I know this might be a little bit difficult to read, but I will just describe what's going on in this little phylogenetic tree here. So this is what the phylogenetic tree of orthocoronaviruses, which is pretty much the same as coronaviruses, and how they have evolutionarily diverged over time. I had to crop off some of this, as you can see, because it was a little bit larger than that, and it would have been too much to put on the slide, and it would have been even harder than it is to read already. So I just cropped off the important part, which is right here. As you can see, SARS-CoV-2 is a descendant of the sarbecovirus, which is a subgenus of Coronaviridae. As you can see by this graph, a lot of recombination occurs. These are all different viruses and just coronaviruses, and this is beta alone and alpha, and you cannot see gamma or delta, but you can see that the recombination happens a lot, and these are all unique viruses. Many of these viruses are present in non-human animals, as you probably can't tell, it's hard to see, but the ones that are colored blue or black are in non-human animals, and the ones that are in red are human viruses. And this is where the term spillover comes into effect. If you haven't heard of the term spillover, spillover occurs when the virus transmits from non-human animals to humans, which is what happened with SARS-CoV-2. It transferred from, they're thinking it's a bat, it transferred from that non-human animal source into humans. And as you can see here, SARS-CoV is also a sarbecovirus, and it's similar but very different in terms of how it has changed and turned into SARS-CoV-2 today. So some information on the structure of the virus itself. It is a single-stranded positive RNA virus Positive sense RNA acts like mRNA and can be translated by the host ribosomes, whereas, say, it was a negative sense RNA, it must carry RNA polymerase within the virion to reproduce. The name coronavirus, it literally means crown. Hence, these little structures kind of look like a crown if you look at it a certain way. And what makes this coronavirus itself unique between other coronaviruses is its spike glycoprotein, which is this little mushroom-looking thing right here. And it's unique to pretty much every coronavirus. So the spike glycoprotein binds to ACE2 in the human body to gain entrance into cells. And if you're not aware, ACE2 is an enzyme that's attached to cell membranes of cells, especially in the lungs, arteries, and etc. Hence why COVID-19 can cause ARDS, or acute respiratory distress syndrome, according to Acosta and Singer in 2020. And what's even more unique about SARS-CoV-2 is the furin-like cleavage site on the spike glycoprotein, which is absent in coronaviruses of the same clade, which was found by Coutard et al. In 2020. Furin is a surface proprotein convertase, PC, and it activates the precursor proteins such as glycoproteins, which is what we are focused on, these little glycoproteins on the coronavirus surface. Furin especially has the potential to cleave viral envelope glycoproteins, thereby enhancing viral fusion with host cell membranes. This furin-like cleavage site has been seen in infectious bronchitis virus, IBV, and had higher pathogenicity. So this furin-like site that is present on SARS-CoV-2 makes it more pathogenic than the previous SARS-CoV, for example. And the structure of it is an envelope virus. It has this envelope covering and protecting the RNA that's contained within. So to talk about the reproduction processes that's important to infection and pathogenicity. So starting from this upper left area, the virus entry entails binding the ACE2 receptors and cleavage by the serine protease TMPRSS2, which is this little thing in green, and allows fusion with the host membrane. Other cellular proteases, for example, furin, which is this orange one that I mentioned in the previous slide, facilitate pH-dependent entry through the endocytic pathway. The predominant entry routes are cell type specific and dependent on availability of select proteases. In the middle here, following uncoating and releasing of the viral RNA into the cytoplasm right here, translation of the open reading frame 1a or ORF1a and ORF1ab produces polyproteins PP1a and PP1ab. These in turn are processed by viral proteases encoded by ORF1a to yield 16 non-structural proteins. Formation of the RNA replicase transcriptase complex, RTC, uses rough endoplasmic reticulum, ER-derived membranes. The RTC drives synthesis of negative RNAs. Full-length negative RNA copies of the genome provide templates for the full-length positive RNA genomes. Transcription further produces a subset of subgenic RNA, including those encoding all structural and accessory proteins. And towards the end of this process, the translated structural proteins and genomic RNA are assembled into the viral nucleocapsid and envelope in the Golgi intermediate compartment and are subsequently released by exocytosis to further bind to other cells and reproduce. So to talk more about the genome structure of the virus itself, which is what I focus on over the summer, it is 13 to 15, 12 functional open reading frame ORFs. The virus is constantly changing, and so are the availability of these genomes. Over 20,000 are available for public access on NCBI Virus, which is what I used primarily over the summer to gather my data. And for the genome length in full, it is 29,903 base pairs in SARS-CoV-2. Details of the genome are ascertained because often there are overlapping genes, which was what I found to be difficult when trying to figure out which proteins are present and how long they are at times. For a little bit of background, an open reading frame gene is a portion of the DNA molecule that, when it's translated into amino acids, contains no stop codons. So it just continues. There's no stop codon right here. PL-Pro and 3C-like protease are attractive antiviral drug targets because they are essential for coronavirus replication. And they are these little guys right here, these little arrows. PL-Pro and 3CL-Pro are to process the viral polyprotein in a coordinated manner. So they are very essential to the genome replication process. So in terms of my research, the question I asked myself was, how does COVID-19 mutate? Specifically, how does it mutate in a local population? So my hypothesis was that I hypothesized that most of the mutations will be found in the spike glycoprotein, and this hypothesis came about because that area is more prone to mutations, as it is what binds to ACE2 and gets introduced into the host cell, and that was what I had assumed going into the research. And different genes mutate with different frequencies, so that must be understood, and some genes are able to have more mutations and still be able to function properly, and vice versa. Say there are genes that are essential to the virus, but they are able to have mutations that still function normally, and those are typically more common because it doesn't compromise the function and survival of the virus itself. And it's important that we study these mutation patterns in order to try and predict for possible changes. If we prepare, we can attempt to prevent. I say attempt as in it might not be successful, but it's a good start. And so that we know what to focus on. There are more areas of more variability. So my methods are, this is my overall process. It was kind of a 3-step process in a way. So I started with gathering my data, which I chose Minnesota COVID-19 genomes, and I aligned them with a reference sequence using NCBI Virus, as previously mentioned. And these mutations were documented into a large Microsoft Excel spreadsheet, and the mutations were recorded and gathered for each genome specifically. For data analysis, the percent commonality of mutations was calculated for each mutation found, and the type of mutation was defined: conserved/non-conserved, missense, silent, nonsense, which I didn't see any nonsense mutations. And whether or not the mutation affects the protein folding was also noted, and that was in regards to the type of mutation. If it was conserved, then I would say the protein folding didn't change as much, versus non-conserved, where it changes to a very different amino acid. I would assume that the protein folding was affected. And the last step was the organization of data. The total mutations per nucleotide were documented and put into a histogram, which you will see in my results. And the total mutations per protein, and I say protein as in like the larger open reading frame proteins rather than each individual non-specific protein. And these mutations were documented and put into a pie graph. So for the data gathering, alignments were made with the Minnesota COVID-19 genomes and a reference sequence, which was this number listed here. And I chose this genome as a reference because it was one of the first ones that was referenced and made, and that I found on NCBI Virus, and other articles that I found have used that as a reference sequence as well because it's the most complete one. And the tax ID that I used for searching up these viruses was the beta coronavirus, and it was 694002. And here are just like some examples or some background info on what I used in my search. I used complete nucleotide sequence and not partial. And the release date was from the 1st of January 2019 to present, just to narrow out some of possible the SARS-CoV genomes from way back in 2002, 2003. I selected this reference strain here that my cursor is on, and then I selected individual Minnesota genome sequences, which were there were 40 that I did in total. And I aligned them using the align tool, and then I zoomed in on each mutation and noted the location, the type of mutation, and the change in amino acid, if any. And again, this sequence was chosen because it was one of the first ones to be sequenced. And then again, I documented this and put it into a Microsoft Excel sheet. For my analysis, details about the strains and their mutations were recorded in Excel, and the commonality of the mutations was calculated after all genomes were aligned. So this is just an example, a little snapshot of what each genome would look like on Excel. It would have the sequence name, the release date, the location, which is always Minnesota, the GenBank number, and I would have the proteins here, the nucleotide positions, and I would just put the nucleotide mutations normally like this, and then I would put the protein mutation itself in parentheses and note the type of mutation and how common it was in the 40 genomes that I looked at. I chose to focus on Minnesota, one, because I was curious to see how it spreads into a more local population rather than like countrywide. And two, it's where most of us in this class reside, so I was just curious how I kind of have a soft spot for Minnesota, so that's why I chose Minnesota. I did not include missing nucleotides in these mutations, as you cannot really determine what mutations occur there, if any, or if it's just a missing nucleotide from the PCR process. And this top image here is a screenshot from NextStrain, which is a website that gives information on COVID-19 genomes and mutations by their locations, by country, and how they've diverged over time. As you can see, COVID-19 mutates a lot, a lot, which is why it's important to understand the genome itself and how it mutates and spreads. And for organization of data, the histogram of the mutation distribution was created on Excel, and the pie graph of percent mutations per protein was created to visualize which protein had the most mutations. And here are my results. So overall, the open reading frame 1a and b contain the most mutations based on the sequenced genomes, followed by S, which is a spike glycoprotein, N is a nucleocapsid, M is membrane, and E is the envelope protein, which E had no mutations at all. And as you can see, ORF1a had 78, B had 65, and so on and so forth. And honestly, I was not very surprised by these results because the ORF1a and 1b are much larger proteins than, say, the spike glycoprotein, which means longer genes equal more opportunity for mutations. So that's kind of what was expected, but it was interesting to see the distribution of all of it and how the envelope protein had no mutations from the genomes that I sequenced personally. So here's my histogram that I made. It might be a little bit difficult to see. I tried to make it as big as possible, but these green bars here represent the number of times the mutation appeared in the 40 genomes, and the numbers on the top represent the number of appearances, little number right here. And the x-axis represents the nucleotide position in intervals of 1,000 nucleotides from 1 to 29,903. And based on these results, you can see that some mutations are more common, and that could be due to random chance via the selection process that I chose. but more research would need to be done on that, and I would have to analyze more genomes to see if that were the case. And as a comparison, this schematic down here is just the COVID-19 genome itself, so you can line up and see where the protein ends, so you can see where the mutations lie. And again, this is just what the virus itself looks like, and this is the genome itself. And further investigation would need to be done to see how the mutations that I found would affect the protein folding and function. So that's just as a little disclaimer that I can't really confirm or deny how this affects the protein folding. So for my discussion and conclusion, what is known is that mutations happen very often in coronaviruses, and this occurs at the replication step, of course, when the subgenomic RNA is generated. This, in turn, increases the homologous recombination rates among these closely related genes, as found by Su et al. In 2016. And these mistakes in RNA copying by RNA-dependent RNA polymerases is what causes these mutations and what causes them to go on from replication to replication. Genomic variations among populations are associated with mortality rates, which was found in an article by Toyoshima et al. In 2020, which found that a specific mutation was associated with a higher mortality rate in those that were infected by this form of the virus. And sadly, I couldn't really determine if any of the mutations that I found were associated with a higher mortality rate, because that data, of course, is not available to the public, which is understandable, but was something I was hoping I would be able to determine. So I cannot say whether or not my mutations are that serious and that it causes higher mortality rates. So that would be interesting for further research, if possible, down the line. So what is not known is that I really could not find any sources specifically addressing mutation similarity specifically among local populations. I found articles that were talking about just the rates of how people got infected, whether it was through public contact, through like being in a dorm type setting and stuff like that, but nothing was really found on the mutation specifically in a population. Genome classification based on patients' likely exposure was, again, imported from other locations, local infections, etc. And the article that I found that on was Liu et al. In 2020. And can we really determine the main point of infection in a local population, i.e. one singular person? Or is this impossible due to the high recombination of coronaviruses, hence why it was difficult, and they still haven't really figured out the origin of the virus itself, like which animal specifically. So can we really determine the main point of infection in a population is the question that I was left with after I finished my research. So what do these results mean? The results of my study explain how COVID-19 spreads in a local population, and the fact that mutations were shared among local genomes is very interesting, and it makes me wonder if one specific person was the host in a way to all the other genomes that I sequenced and analyzed. Yeah, this could be due, again, to random chance, or more likely the strains analyzed were from cases that had the same person infecting them. And again, I could not see who they belong to, so I cannot determine if what I previously mentioned is necessarily the case. What was not expected is how evenly distributed the mutations were. Initially, I hypothesized that most of the mutations would be located on the spike glycoprotein, as that is what makes COVID-19 so dangerous and how it mutates on the spike protein. This statement itself is in line with studies done by Coutard et al., which show that often the mutations are found on the S protein. And again, if I was to look back at my pie graph, you could see that there were a lot of mutations on the S protein, but of course, the ORF1a and B had more mutations because of just the fact that they're larger proteins overall. And the frequent mutation of the virus can make it difficult to create a vaccine, which further emphasizes the importance of knowing where the virus mutates and with what frequency. And if we know what parts of the viral genome mutate more often, we can find areas of homogeneity, which could be used as possible areas for vaccine targeting. Future directions. As this work was, of course, done exclusively from my laptop due to COVID, I learned that my laptop is not strong enough to handle working with larger files, and I discovered this when trying to make the proteins on a program called FoldIt, which you may or may not be familiar with. And I was not able to see how the protein mutations affected the energy of the protein as well as the specific folding. My future goal is to use on-campus desktop computers to build these proteins and see just how much or little the proteins change before and after the amino acid mutations. And I would also like to analyze more than the 40 I did for this poster. I was initially not expecting it to be as time consuming, but it was very time consuming, more than you would think. So I only really had time for 40, but I would like to do more to get more accurate results that are more applicable rather than just 40. And thank you for listening to me talk about my research. And feel free to ask any questions, and I will try my best to answer them. I would like to thank my advisor and pretty much the person that held me together this summer was just Dr. Irina Makarevich. She was the one that helped give me the idea for this presentation. And give a little shout out to Alicia Palacios, who was also doing SARS-CoV-2 research, but on a little bit of a more different topic, so you'll hear from her later. She's super awesome, and she's very helpful over the summer as well. I'd like to thank all my family and friends that heard me talk about my research all summer. It's literally all I ever talked about and everything like that. So I'd like to thank them for listening to me and helping me out when I needed it. And again, thank all of you for listening to me talk for this seemingly long period of time. And as a reminder, wear your mask, wash your hands, and social distance, because if my talk wasn't enough, yeah, you just got to do it. Just do what the CDC says, because they know what they're talking about. Thank you.